Hello, good afternoon, everybody. So it's 3 o'clock. Thank you for joining this latest Asphalion webinar. So I'm John Giblin. I'm a Scientific and Regulatory Affairs Officer at Asphalion, and I'm going to be talking about the latest developments in vaccines to prevent COVID-19. So just to give you an overview of the topics that we're going to cover today, firstly, we'll start with an introduction, and then we'll talk about why we need a vaccine, and after that we'll talk a little bit about historical vaccine development, and then we'll move on to the main part of the webinar today, which is to explain to you a little bit about the types of SARS-CoV-2 vaccines in development. And then to close the webinar, we'll talk about the clinical development of SARS-CoV-2 vaccines. So by way of an introduction, as I'm sure you're probably all aware, COVID-19 is a disease caused by a new strain of coronavirus. So although it's a new strain of coronavirus, these types of viruses have been described since the 1960s, and they take their name from, when you look at them under the electron microscope, they have kind of a characteristic halo, which when they were first discovered was compared to the corona of the sun. So that's where they get their name from. And the causative agent of COVID-19 is SARS-CoV-2, the SARS-CoV-2 virus, which stands for Severe Acute Respiratory Syndrome Coronavirus 2. The first case was reported from China to the World Health Organization at the end of last year. And as of today, there have been more than 4 million confirmed cases, resulting in nearly 300,000 deaths around the world. So, as I mentioned, SARS-CoV-2, which causes COVID-19, is one of 7 human coronaviruses. And these viruses are known as zoonotic viruses in that they originate from animals. And in the case of coronaviruses, they usually derive from bats, as we can see here. So of the 6 human coronaviruses that were described before SARS-CoV-2, 4 of them appear to have been derived from bats. They sometimes pass through an intermediate host before they go on to infect humans. Okay. Fortunately, 4 of the 6 previously known human coronaviruses, so HCOV-NL63, HCOV-229E, HCOV-OC43, and HCOV-HKU1, cause mild respiratory and gastrointestinal illnesses, which are similar to the common cold. However, in the recent past, there have been 2 serious outbreaks of novel coronaviruses. The first of these occurred in 2002 and ended in 2004, and this was the SARS outbreak, which stands for Severe Acute Respiratory Syndrome. This was caused by the SARS-CoV-1 virus, and it was largely localized to China and the Far East, and resulted in over 8,000 cases and nearly 800 deaths. The second major outbreak of a novel coronavirus before this one occurred in 2012, and this was MERS, Middle East Respiratory Syndrome, which was caused by the MERS coronavirus. And this is actually technically an ongoing outbreak. There are still cases observed every year since 2012. And this has resulted in over 2,500 cases and nearly 900 deaths. So SARS-CoV-2 was identified as being associated with a pneumonia outbreak in Wuhan in China. So the genome sequence of the virus was first published just over a month after the first reported case at the end of last year, and it was the virus was isolated from the bronchoalveolar lavage fluid of a worker that worked at this market in Wuhan, China. And from its sequencing of its genome, it was placed in the family tree of coronaviruses, and it's, as you can see here, it's very closely related to coronaviruses that are found in bats, and more distantly related to the causative viruses of the SARS outbreak in 2002, SARS-CoV-1, and the MERS outbreak of 2012, MERS-CoV-1. So just to go into the features of the SARS-CoV-2 genome a little bit, because it's relevant for vaccine development. So SARS-CoV-2 is a single-stranded positive sense RNA virus. Its genome encodes 16 non-structural proteins, which are denominated NSP1 to 16, and they're involved in the suppression of the host immune response, transcription of the genomic material of the virus, and replication of the viral genome. There are also 6 accessory proteins which are thought to contribute to the pathogenesis of the virus, but as of yet they're poorly understood. The most interesting from the point of view of vaccine development are the 4 structural proteins. So these are the spike glycoprotein, also known as the S protein, which is involved in helping the virus latch onto cells to infect them. The matrix protein, the M protein, which is a protein found on the surface of the virus, as is the envelope protein, another surface glycoprotein of the virus. And then we also have the nucleic acid protein, which encapsulates the genetic material of the virus. So it's a bit slow to start moving. Right. Excuse me. Sorry. Okay. So SARS-CoV-2 is quite genetically is already becoming quite genetically diverse. So sequencing of SARS-CoV-2 from isolates taken from patients around the world, over 30 countries, have revealed that there are several high-frequency sites for mutation, and this may have some implications for vaccine development. So as you can see in the left panel, the ancestral strain, which was isolated from patients in Wuhan, China, has already been started to introduce changes in its genome. So you can see that cases in patients from Australia, you can already see that there's been changes introduced into the genome, and you can also identify where a viral strain of SARS-CoV-2 has come from by changes in its genetic sequence. And of interest, several of the proteins that are involved in on the surface of the protein are also quite prone to these high-frequency mutations. So we show here there's positions in the spike protein that are also prone to mutation, and also in the nucleic acid protein. So let's talk now about SARS-2 infectivity. Excuse me a moment. Yes, sorry, I just missed a slide. So I just wanted to explain as well a little bit about the SARS-CoV-2 life cycle and its characteristics. So SARS-CoV-2 is transmitted by, as you probably all know by now, by direct contact through droplets or aerosols, and also by indirect contact for example, contact with a contaminated surface following touching the eyes, nose, or mouth. Once it gets into the body, the virus enters cells by attaching to this receptor, angiotensin converting enzyme 2, which is found on the surface of cells in the respiratory tract, but it's also found on most other cells of the body. So this may explain some of the other symptoms that are not some of the non-respiratory symptoms which are associated with COVID-19. And the virus, just to emphasize, enters the cell via the spike protein. Then it transcribes its genome, packages, makes the nucleic acid protein, the surface protein, the membrane protein, and the envelope protein, packages the virus again, and then is released from the cell to infect more cells. So during this replication process, errors can be introduced, and this is what we see from the sequencing of SARS-CoV-2 isolates taken from patients around the world. So let's talk a little bit about SARS-CoV-2 infectivity, because this has important implications for vaccine development. So the basic reproduction number of a virus refers to the number of cases that will occur in a susceptible population as the result of infection by a single individual. So various viral diseases have different basic reproduction numbers. So we have, from one extreme, we have measles, which has a basic reproduction number of over 10. So for every one person that's infected with measles, they will infect 10 other people. To MERS, which fortunately had a basic reproduction number of less than 1, which probably explains why it hasn't reached epidemic or pandemic proportions. Now, in the case of SARS-CoV-2, the current evidence suggests that the basic reproduction number of SARS-CoV-2 is 2.2. So for every one person that's infected with SARS-CoV-2, just over 2 people will become infected in a susceptible population if no other measures are taken. So the other important number is the effective reproduction number, and this refers to the number of cases that are occurring in the population at any specific time as the result of an infection by a single individual. So when the effective reproduction number is greater than 1, the infection spreads through the population exponentially. When the effective reproduction number is less than 1, the epidemic will eventually stop. So all the measures that are currently being taken by the authorities are with the aim to eventually reduce the effective reproduction number to less than 1. So the social distancing measures that governments have taken all around the world are really aimed at reaching this point. The effective reproduction number can also be attained by the attainment of immunity in the susceptible population following infection or through vaccination. So why is the vaccine required? So if we look at the relationship between the basic reproduction number and the effective reproduction number, we can see that the immunity in the population has an influence on the effective reproduction number. So in the case of a disease like measles, which has a basic reproduction number of over 10, only when there is 90% of the population displaying immunity will the effective reproduction number of this illness drop below 1. And this is one of the reasons why it's so important for the entire population to be vaccinated against measles, because if the level of immunity drops below 90%, then the effective reproduction number will rise above 1 and we'll end up with an epidemic with the disastrous consequences. But going back to SARS-CoV-2, so as I mentioned earlier, the basic reproduction number of SARS-CoV-2 is 2.2. So in this case, for the effective reproduction number to drop below 1, what is required is 60% of the population to be immune. and I've read some estimates that think that maybe say to be absolutely sure it should be of the order of 70–80%. So what is the prevalence of infection in the general population? So one thing that the public health authorities were thinking is that maybe if a sufficient proportion of the population have already been infected, and assuming that once they're infected, they have an immunity that lasts for a significant length of time, then maybe we've reached a herd immunity threshold, which would be 60%. So several studies have been undertaken to look at the prevalence of infection in the general population. And I'd just like to emphasize one of the studies that was conducted by the IS Global Barcelona Institute for Global Health. And they looked for antibodies against SARS-CoV-2 in healthcare workers at the hospital clinic in Barcelona. And they analysed nearly 600 healthcare workers, and they saw that 54 of them tested positive for antibodies. And interestingly, approximately half of the subjects that tested positive for antibodies did not experience any symptoms. So this is another problem with checking the spread of the disease, is that there are many asymptomatic carriers of the disease, which makes it very hard to isolate the cases of the disease. And in total, 11% of the workers displayed evidence of past infection by the presence of antibodies, or current infection by detection of genomic material of the virus by PCR. Now, given healthcare workers are a high-risk group, it's probable that the prevalence of SARS-CoV-2 antibodies in the general population is even lower than 11%, and very far from the 60% herd immunity threshold. And as was published yesterday by the Spanish Science Ministry and the Health Ministry, that prediction has been shown to be correct. So nowhere in the general population is the prevalence of infection higher than 15%. So we're very far from the 60% herd immunity. So I think it's clear that a vaccine is required. So maybe just to emphasize the point, here's a quote from Peter Piot, who's probably the most famous living virologist at the moment. And he has basically said, let's be clear, without a coronavirus vaccine, we will never be able to live normally again. So the only real exit strategy is a vaccine that can be rolled out worldwide, which means producing billions of doses. So it's an enormous challenge in terms of manufacturing logistics. So before we talk about the vaccine development and the different types of vaccines in details, I just wanted to go through very briefly some of the concepts of the immune response. So in the context of SARS-CoV-2. So the immune responses can be divided into two kind of phases. So there's an early phase of which occurs between the time of infection and 12 hours after infection, which is the innate immune response. And this is the response of the immune system to the recognition of non-specific viral components. So for example, viral DNA or RNA, for example. And then later on, in the days following the infection, the adaptive immune response becomes important. And so the adaptive immune response involves B lymphocytes, which produce soluble antibodies against the virus, which circulate in the circulation, so the humoral response, and also the cell-based response, which is mediated by T lymphocytes, which recognize and destroy infected cells through binding to a specific viral epitope, through recognizing a specific viral antigen. So just to emphasize, sorry. So just to emphasize the development of adaptive immunity to SARS-CoV-2. So the virus is released from infected cells. It's ingested by dendritic antigen presenting cells, which then basically chop up the virus and then display peptide antigens on their surface. These peptide antigens are then recognized by T cells, which can then promote the clonal selection and proliferation of B cells, which secrete antibodies against that peptide antigen, and also the production of cytotoxic T cells, which, as I mentioned before, can recognize and destroy infected cells. And the principle of vaccination is that the principle of immunity after the infection is that there are memory B and T cells which can potentially provide immunity for months or years after the infection. What we don't know, and as I mentioned earlier, is we don't know for SARS-CoV-2 whether there is immunity after an infection, and if there is immunity, how long that lasts for. Next slide. So the principle of vaccine development is that the aim of the vaccine is to expose the body to an antigen that won't cause disease while provoking an immune response that will be able to block or kill the virus if a person becomes infected with the virus at a later date. Okay, and so this involves both responses. So ideally, the vaccine should activate both arms of the immune response. So it should activate adaptive immunity via a specific antigen which targets the virus, and it should activate innate immunity via non-specific danger signals, which are usually achieved by the addition of an adjuvant, which is then combined in the vaccine, and then the vaccine produces the immune response, either a humoral response, which produces antibodies which may block infection, or a cell-mediated response by T cells, which recognize infected cells and kill them. The exact type of response required for a SARS-CoV-2 vaccine is not clear at the moment. So just as a way of a historical overview of vaccine development, so it's one of the success stories of medicine historically, that there, as you can see on the left panel, there are many diseases that were responsible for the deaths of millions and millions of people, that now there are vaccines available, and that we take for granted that these diseases, which are highly lethal, we have protection against them. We can also see that vaccine development has been faster or slower depending on the type of illness. So, for example, dengue fever was first described in 1907, and only in 2019 a vaccine became available. And there are also, should be pointed out, that there are also many viral illnesses that vaccines are still not available. So, for example, the HIV virus, mononucleosis, and the Zika virus. So, just as a way of emphasizing the scale of the challenge that we all face at the moment is the timelines for vaccine development. So this is an industry average for vaccine development, and so it goes through the typical stages of development of a product. So identification of the antigens, produce the antigens, then preclinical testing in appropriate animal models, then phase 1 studies to look at safety and initial immunogenicity, moving into phase 2 studies with larger populations of patients, and then phase 3 studies with basically showing that the vaccine provides protection against the particular illness. And this process typically takes 10 years. So really, we're hoping to have a vaccine within 18 months. So it really is an amazing challenge that we face. So let's start to talk about the development of a SARS-CoV-2 vaccine and talk about the different types of vaccines and some of the interesting products that are in development at the moment. So as we can see here, there's a website with the World Health Organization which is constantly being updated with vaccines which are in the process of development. And as of the 12th of May, so as of a couple of days ago, there are 8 candidate vaccines in clinical evaluation at the moment, and there are 102 candidate vaccines in preclinical evaluation. And when I looked at this 2 weeks ago, I think it was like 4 candidate vaccines in clinical evaluation and 70 in preclinical. So it's almost it's growing at a very fast rate, the number of products in development. So several types of vaccines are under development, and they can be categorized according to the platform through which the antigen is introduced into the body. So we have whole virus vaccines, viral vector vaccines, protein-based subunit vaccines, virus-like particle vaccines, and nucleic acid vaccines. So we'll go through these one by one and I will try to emphasize the key points and some of the products that are in development at the moment. So first of all, we'll talk about whole virus vaccines. So these vaccines are developed either using an inactivated form of the virus or a weakened form of the virus, which is also known as an attenuated virus. So this is one of the most successful historical approaches to vaccine development. So the same approach was used to develop the measles virus, which is a attenuated, weakened virus, and the polio virus, which is an inactivated virus. And just to bring your attention to 2 products which are currently in development. So there's Picovac, which is a SARS-CoV-2 vaccine based on the inactivated virus, which is being developed by Sinovac Biotech, which is a Chinese biotechnology company, and that's currently in the phase 1 to 2 trial, phase 1 or 2 trial to look at safety and immunogenicity. And there's another product in development, an attenuated virus, CDX-CoV, which is being developed by the American firm Codagenics in collaboration with the Serum Institute of India, which is one of the largest manufacturers of vaccine products in the world. So just to give you an overview of some of the pros and cons of this approach to vaccine development. So in the case of both inactivated and attenuated viruses, as I mentioned earlier, these are proven platforms. So there are existing licensed products. Some of the drawbacks, well, some of the cons with these products is that, particularly with the inactivated virus, it requires the handling of large amounts of infectious virus. So, for example, with the Sinovac product that's been in development, I've seen that basically they take isolates from patients and then they grow them through cells to make large quantities of the virus, and then they inactivate the virus before constituting the vaccine. So obviously it represents a technical challenge to ensure that the virus doesn't escape or pose a threat. Also, in the case of inactivated viruses, because they go through a chemical process to basically inactivate them, confirmation of antigen and epitope integrity needs to be confirmed. Similarly, for attenuated or weakened viruses, one particular issue with SARS-CoV-2 is that due to its relatively large genome size, creating infectious clones for an attenuated version of the virus is a relatively slow process. So with an attenuated virus, the idea is that the virus is passaged through cells and mutants are selected that have lower pathogenicity and lower infectivity. And this is a process that can take time if the genome size is rather large. Extensive safety testing is required, and phenotypic or genotypic reversion can still cause some disease. Viral vector vaccines. So these can be categorized into two categories. So replicating viral vectors or non-replicating viral vectors. And in both cases, the principle is similar. It's analogous to gene therapy, but instead of delivering a transgene, an antigen is delivered. So in the case of a replicating viral vector, these should be emphasized that these are viruses that are not SARS-CoV-2. These are other types of viruses, but they carry a gene from the SARS-CoV-2 virus. So in many cases, this is the gene for the spike protein. And as they can still replicate, they infect cells and replicate. And this has the advantage that they mimic an infection, which produces both T and B cell responses. And just to draw your attention to a SARS-CoV-2 vaccine based on this technology that's in development, is a collaboration between the Institute Pasteur, Themis, and the University of Pittsburgh Center of Vaccine Research, also with support from the Coalition for Epidemic Preparedness Innovations. And they're developing one of these replicating viral vaccines based on an attenuated form of the measles virus. So this is just brief. I haven't prepared a slide, but I just briefly wanted to mention the Coalition for Epidemic Preparedness Innovations. So this is a consortium of donors, which basically was formed in Davos in Switzerland in 2017, and they basically are supporting many preclinical and clinical development programs of SARS-CoV-2 vaccine, of SARS-CoV-2 potential vaccines. So they're also, I think one of the donors is the Bill Gates Foundation. So they're investing a lot of money in some of these vaccine programs. And also should be noted that it's not just SARS-CoV-2 vaccines that they support. They also support many of the other vaccines of infectious diseases around the world. So until recently, there were no approved products for this type of platform. But in the last couple of years, there's been two of these replicating viral vaccines that have been approved. So one is Ervebal, which is a vesicular stomatitis virus expressing a surface glycoprotein of the Ebola virus, and Dengvaxia, which is an attenuated yellow fever virus, which expresses surface proteins of the dengue virus. So this is another technology that's proved and has been shown to produce products that can be used. The second subcategory of viral vector vaccines are non-replicating viral vectors, and these are generally based on non-human adenoviruses, so often primate viruses, so from chimpanzees, for example. And the reason for this is to elude pre-existing human immunity. So adenoviruses are endemic in human populations, and many people in the population have some immunity to these viruses. So it would not be very useful to have a vaccine packaged in an adenovirus that basically produced an immune response against the vaccine and destroyed the vaccine. So for this reason, non-human adenoviruses are typically used in this platform. Just to mention, because in gene therapy, another subclass of viruses, adeno-associated viruses, are used, and I haven't come across any of these any products in development with adeno-associated viruses to date. But it's worth noting that in the previous outbreak of coronavirus, the SARS outbreak with the SARS-CoV-1 virus, preclinical studies were conducted with the use of adeno-associated viruses to administer antigens via intranasal administration. But going back to the current outbreak, so CanSino Biologics and the Beijing Institute of Biotechnology are initiating phase 2 trials in China with a non-replicating viral vector to evaluate safety and immunogenicity. And also a trial that's received quite a lot of media attention in the last few weeks is a trial from the University of Oxford with the support of AstraZeneca, which is a chimpanzee adenovirus-based vaccine, which is currently in a phase 1 clinical study, which has been initiated to assess efficacy and safety. And I think they're planning to move into phase 2 studies fairly quickly. Another major project is with Johnson & Johnson, who have started a phase 1 clinical study, which they plan to start in September 2020 and hope to have data on safety and efficacy at the end of this year. So these types of vaccines have a couple of advantages that should be emphasized. So no infectious virus needs to be handled, and they also produce an induction of high cellular and humoral responses. And as I mentioned, pre-existing immunity to the vector may affect the vaccine effectiveness. So the third type of vaccines are subunit or protein-based vaccines. And probably of all the vaccine types, this is the most common type that's in development. So there are currently around 26 in development, and they're mostly based on the spike protein, but with a few with the M protein, which are both surface proteins of the SARS-CoV-2 virus. And the rationale for this is that they've seen that neutralizing antibodies from recovered COVID-19 patients were found to target these proteins. So the hope is that by using these proteins as part of a vaccine, immune responses will be induced to produce neutralizing antibodies, which will prevent the infectivity of the virus. So these types of protein-based vaccines are often injected with an adjuvant to enhance the innate immune response. So the protein itself is there to activate the adaptive immune response, and the adjuvant is there to activate the innate immune response. And just to emphasize a product that's in development at the moment, so Sanofi Pasteur and GlaxoSmithKline are developing a vaccine based on the S protein, which is made by insect cells infected with a virus which specifically infects insect cells, the baculovirus. And this is based on technology that's already used to produce the approved seasonal influenza vaccine, Flucelvax Tetra. And they're hoping to begin phase 1 trials in late 2020. So some of the advantages of these types of vaccines is they can induce strong cellular and humoral responses if administered with an adjuvant, and high titer neutralizing antibodies to specific regions of the virus can be potentially induced. Again, antigen and epitope integrity requires confirmation. There are some issues with global production capacity for production of these proteins, which is associated with a high cost. So the fourth type of vaccine are virus-like particles, and these are based on purified recombinant proteins which are found on the surface of the virus, which are then assembled into a virus-like particle. And there are currently 4 of these in development for vaccines against SARS-CoV-2, and it's a proven technology that these types of vaccines are already authorized against hepatitis B and human papillomavirus. And just to draw your attention to one of the interesting products that's in development, so Medicago Incorporated, which is a developer of plant-based VLP vaccines for influenza, have a candidate for SARS-CoV-2 in preclinical development. And their system involves introducing the recombinant viral genes, so the genes which correspond to the viral surface proteins, into a bacteria, which then infects plants, and then the plants synthesize the virus-like particles. So basically, they mash up the leaves and then purify the virus-like particles that then can be introduced into the vaccine. And then the final type of vaccines that I mentioned are nucleic acid vaccines. So these have received a lot of attention in the media recently because they're the fastest vaccines to be developed. So Inovio are developing a DNA vaccine, and this is a company that also has a record of developing DNA vaccines against HIV, Ebola, Zika, Lassa fever, and MERS viruses. And they're currently in phase 1 clinical studies to investigate the safety profile and immunogenicity. So one feature of DNA vaccines is that the DNA vaccine encodes a component of the virus that is considered most likely to produce an immune response. So in this case, again, the S protein, the spike protein of the coronavirus. And then to administer the vaccine, the vaccine is administered intradermally by a specialized device, which basically is loaded with the vaccine and then delivers an electrical current, which basically opens pores in the cells, allowing the DNA plasmid to enter and then to transcribe the mRNA and translate the protein, which hopefully can induce the immune response. So as I mentioned, these are rapid to develop, easy to scale up, and with low production costs. And also DNA vaccines generally have a high stability. One of the drawbacks is that a specific delivery system is required, and also there are no products currently approved. I think we mentioned that there was one that was used to introduce vaccines into salmon or something like that, but apart from that, nothing for humans. So the RNA vaccines, the principle is the same. So the nucleic acid forms the vaccine, is introduced, and then produces the viral proteins once in the cell, and then hopefully these viral proteins produce the immune response. And one of the most headline-grabbing trials that's underway at the moment is the one that's with Moderna, in collaboration with the National Institute of Allergy and Infectious Diseases in the United States, and they have an mRNA vaccine against SARS-CoV-2 in development. And they currently have a phase 1 trial underway to assess safety, reactogenicity, which is the exaggerated immune response, which is undesirable, and immunogenicity. And these mRNA vaccines, they don't require specialized administration devices because they can be encapsulated in lipid nanoparticles and then administered by injection. And just on the subject of manufacturing, we can see that the United States government have funded Moderna and Lonza to make 1 billion doses of this potential vaccine. So they already have the capability in place. If this vaccine proves to have efficacy, they already have the capacity to scale up the production. The second RNA vaccine that's also received a lot of attention, which is being developed in Europe by a European company, is the mRNA vaccine BNT162. So BioNTech, the German company, in collaboration with Pfizer, are testing 4 of these mRNA vaccine candidates, and their phase 1 and 2 trial is currently underway to assess safety, tolerability, immunogenicity, and potential efficacy. And this is also a lipid nanoparticle encapsulated mRNA vaccine. So as I mentioned, the same for the DNA vaccines, rapid production and development is possible. The only thing is that the technology, I wouldn't say it was unproven, but there's no approved products. So it's something where hopefully they will prove to be successful. So just to summarize the phases of vaccine development and the context of the normal situation for vaccine development and the context in the emergency situation that we find ourselves in now with SARS-CoV-2. So from preclinical testing, the test of antigens, the test of adjuvants in animal models, also using information from other related viruses, could typically take 5 to 10 years. Then the clinical testing phases as well, rather long length of time. And then also the monitoring, which is also an important aspect in phase 4, because in mass vaccination, if there are adverse events which only occur in 1 in 100,000 people, maybe if you're only administering the product to 100,000 people, it's maybe could be considered an acceptable risk. But if you're inoculating billions of people, then maybe the risk benefits have to be considered very carefully. What's being aimed for in the development of SARS-CoV-2 vaccines is to do all this process in 6 to 9 months by overlapping some of the phases. But also it should be mentioned that there is a wealth of information from previous outbreaks of coronavirus, which have also been used to inform vaccine development of the SARS-CoV-2 vaccine. So information from the SARS-CoV-1 and the MERS outbreaks. There were several vaccines which are still in development, actually, which have also been very helpful to basically, which basically means that the vaccine development hasn't started from scratch. So I just wanted to raise a couple of points just briefly about clinical testing of vaccines. So at the moment, we're in phase 1 and 2 trials. So all the products that are in clinical development that we've mentioned that I've mentioned previously are all in phase 1 or 2 clinical testing. So in these trials, generally, aside from the important safety endpoints, the primary efficacy endpoints are the assessment of humoral responses, so the assessment of B-cell responses, and also the assessment of the antibodies that are being produced. So are the antibodies that are being produced, do they have a neutralization activity in vitro assays? And the second primary efficacy endpoint is the assessment of T-cell responses. So this is the isolation of T-cells from vaccinated participants in the trial, and then a measurement of their cytokine release in response to an antigen challenge. It's important to note that these are not surrogate endpoints. So these endpoints do not give any indication of the potential of the efficacy of the vaccine. So they don't give any indication of clinical benefit. They simply allow the vaccine to go into larger trials with the hope that it will prevent infection by the virus. And so an example that I mentioned here is that the antibody generated may not prevent infection. So the presence of antibodies may not prevent infection. And then I just wanted to make a brief mention of posology as well. So from the point of view of the World Health Organization, ideally a one-shot vaccine would be the ideal. So one shot and that's it. However, in reality, what is probably more likely that will happen is that prime boost immunization will be required. So more than one immunization will be required, and perhaps with 2 different types of vaccine. So for example, an initial immunization with a DNA vaccine, followed by a viral vector-based vaccine. And the spacing between administrations may affect both immunogenicity and safety. And to facilitate this process, the World Health Organization have set up this solidarity vaccine trial. So this is a large international multi-center trial with a core protocol, which basically is designed to coordinate the evaluation of the safety and efficacy of SARS-CoV-2 vaccines. So one thing that is clear with the number of the number of SARS-CoV-2 vaccines in development. Not all of them can go through a full clinical development program. So I think what's very important in the current situation is that viable candidates are identified very, very quickly and moved on, and less viable candidates are eliminated without going too far down the road of development. So this is the idea of this solidarity vaccine trial. So it's a centrally randomized trial with a common placebo group, and it has an adaptive design. So this is what I was saying, that so basically candidates are added as soon as they become available, and others are eliminated if there's no benefit seen relative to placebo. So the aim is to, within 3 to 6 months, that we get some idea of which candidates are viable and which candidates are not viable. The primary outcome is virologically confirmed COVID-19 disease. So the disease rates from participants receiving each vaccine will be compared with disease rates for the concurrently randomized placebo and control recipients. Secondary outcomes are immunogenicity, immunological correlates of risk, so overreaction to the immunization, viral shedding, transmission patterns, and efficacy after first dose. And safety, the possibility of enhanced disease has to be monitored as well. If the vaccine meets the success criteria, new efficacy and lack of benefit criteria will be introduced. So just finally, another concept that has been introduced in the potential clinical development of SARS-CoV-2 vaccines is the concept of challenge trials. So these are trials that similar studies have been conducted with malaria and influenza. So this is the idea that rather than waiting for the participants to become infected, basically you challenge the participant with the virus. So this is obviously something that ethically has to be considered very, very carefully. And I've just put some headlines for your general interest, and if anyone's interested in delving into the ethical and technical aspects of this, there's some documents from the World Health Organization that you can look at. And I think with that, I've almost used up the hour, so I think I'll thank you for your attention, and just bring your attention to the next webinars in the series. So on the 21st of May, which is next week, Dominique Montferrer is going to give a really interesting webinar about the medical devices regulation postponed, an opportunity that cannot be missed. And then at the end of this month, Lydia Canavas is going to give a great talk about expedited regulatory pathways. Thank you for your attention, and if anyone has any questions. Oh, sorry. Yeah, I was answering the questions by writing, but I think if you can still hear me, Rosanna, in terms of the type of vaccine that I think would first reach the market, I'm not sure, but maybe one of the nucleic acid vaccines, probably just because of how rapidly they're being developed. Probably one of those would maybe reach, at least from the point of view of an emergency use for health workers, would maybe be one of the most feasible ones that would reach market first. I think maybe in the long term, perhaps the more traditional viruses will also eventually get to the market, but I don't think they will reach the market anytime soon. I think one of the things that I didn't mention in the talk, but also I think it's important that there's more than one virus reaches the market, because I think there's going to be issues with initially. So I think there might be some vaccines that will be used for use in health workers and in vulnerable populations. whereas others that will maybe be more useful for the general population. But thank you for your question. I'm just looking at another. In terms of feasibility and economics, I'm not really sure. I mean, I think it's not really so much my area of expertise, but I think that, again, if the nucleic acid vaccines are shown to be efficacious, I think they're very feasible, because the production can be quite rapid, and so it can be distributed very quickly. And another question. Oh, sorry, Lewis, I'd written you an answer to the question. Yes, so that in terms of the spike proteins, there could be, if it's mutating very rapidly, then that will be an issue for future vaccine development, I think. And it's one of the things where I think there's a kind of assumption that the S protein is the most appropriate target, and that, to be fair, is based on previous studies with the SARS and the MERS viruses in the future, in the past. So that answers your question. And hello, Trudy. In terms of the PowerPoint, I think it will be. I think we will. We'll make it. It's going to be available on YouTube, I think. So it will be available. It will be available from there. And I'll consult with. So maybe it will be available for download as well. I'll have to ask. But thank you for your question. Anybody else have any other questions? Okay. I think in that case, then, if no one has any other questions, I will disconnect and wish you all a happy afternoon, and everyone stay safe and healthy. Thank you very much for listening. Thank you.